Him got opened a little bit prematurely, and so we are really starting at 6. Hey, Katarina. Here.
Hi, Victoria. How are you?
Good. I'm jumping down to the audience to put you in front. Goodbye. Wait. I can't. There we go. Thank you.
Thank you, Victoria, but it was not necessary, but that's
Yeah,
nice of
yeah.
you. How are you?
That's the right thing to do. Oops. Hey, Jamie.
Oops, sorry, I wasn't muted. How are you, Victoria? Hiya.
Good, thanks. Such a peaceful room.
The
Yeah,
sound
can
of
you
silence.
hear the ocean actually? Or is
Yeah,
it too far away?
I do, now that you say what it is. I like that. That's a great background for a room.
Yeah, I agree. We
Oh, it's
still have some
perfect.
time. Oh, go ahead.
Yeah, it's perfect. Because remember when I was on the beach and it was just too noisy and nobody could hear anything? I like yours.
I was enjoying that UFO music room that both of you were in a couple of days ago. I popped into when I saw you both in there. It was such amazing sounds, so relaxing.
Hi, everyone. You hear me?
Hi,
Hello,
Cliff.
Dr. Cassidy. Oh, hi,
Hi.
welcome.
Thanks.
Yeah, welcome. Thank you for coming. I put your slides up
Yeah.
if you want to check. Everything is working. Thank you.
Okay.
Hello, Clifford. Welcome.
Hi. Thanks.
How are you doing today, Doctor?
I'm good.
Hope the weather's not been too hot for you where you are. It's been Everybody's been getting the heat today.
Yeah, it's been hot here too, actually. I guess I was wondering if you hear the air conditioner going, but hopefully not too bad.
Well, you've got the air conditioning with you, and in Katerina, when she unmutes, she will hear a little bit of the ocean in
Can
the
I make
background.
everyone jealous? It's the last day I'm here. I'll drive home back tomorrow. It's
Where are you?
actually chilly here by the ocean. In the city, it's crazy hot, but I'm in Southampton and on Long Island
Okay,
having dinner by the beach. So it's
nice.
actually chilly. My kids are complaining that it's a little bit cold. So
Okay, well,
sorry.
That's almost like a humble brag, Katerina. I'm eating by the beach, but it's a little bit cold.
Yeah, sorry. But tomorrow, even at night and the next days, I'll be suffering like everyone else. So
hmm
What about
yeah.
yourself, Doctor? What have you been doing today? Sorry.
Sorry, I was just saying I opened the slides. I think they look okay.
yeah, perfect. I hope you enjoying your summer. I don't know if you're doing something fun during the summer, but
Yeah, well, I'm going away in 3 or 4 days. I'm from the west coast of Canada originally. My family's there, so I'll be leaving on Saturday for a couple weeks. And be by the ocean there.
okay, we should have done the room then so you could brag while I'm in the city suffering.
Yeah. Well, it's hard to
Like
get
it's
an
good.
impression with only audio. You don't get the full picture, but yes.
Well, it's kind of impressive how personal these usually there's not scientific rooms like usually it's more kind of discussion rooms and
Mm-hmm.
sometimes personal sharing. How much people share because it's just voice and they feel like I think people feel more comfortable when they are not on video, like
Yeah.
so people share a lot here, don't you think, Victoria, on this app? Like,
Yeah, I do. I just shared a photo of where I'm working these days because Clifford, you were saying audio only. So here's some capability to have a little
Okay.
bit another layer. So it's a river and some Canada geese are there. Jamie, it's a photo of side view of a river with a little bit of the bank and then some trees behind and some Canada geese are on it. It's where I go during my break and take a walk.
That sounds so lovely.
It is.
And the Canada geese, are they friendly, or do they like come and honk at you if you get too close?
Well, I really give them their space. So, I don't know, they could, but I always figure they got here first, so I don't judge. Yeah, I don't know. I'm sure if I approach them, they might want to do something like bite, maybe. But I've never seen them chasing anyone. They just
That's
make
good.
some noise and do some fancy formations in the sky and
Plus,
eat.
they probably came from much further away than us, right?
Yeah,
Are these the ones that like fly across continents and stuff?
they
Those are
are.
amazing.
Yeah, they are. Yeah, it's amazing when you hear them overhead honking too. Because you can hear them approaching and then hear them leaving. It's pretty exciting. Big V formations up there.
We see a lot of them around here.
I don't know, where are you right now?
I'm in Canada, so I'm in Ottawa,
Oh,
which
you
is,
are. Okay. Maybe we've seen the
yeah,
same ones.
they come through, spend a lot of time, and fly off in the V formations. Every winter they fly south.
They come over here to my house. Hey, Sesi Rahim.
Hello. Hi, Katerina. Hi, Jamie. Hi, Victoria. We meet again, Victoria. Just kidding. Hi, Clifford. How are you?
Hi, good.
Welcome.
Thank you.
Sorry, Clifford, we're just sharing the rooms just now and setting everything up. So, but thank you very much for being here. I'm actually quite looking forward to your talk. Um, Incredibly fascinating topic that you've been working on.
Yeah, thanks. Yeah, I'm sure we'll have lots of time to dig into it, and looking forward to sharing. Do we usually start right at the top of the hour?
Yeah, we'll stop at the start of the hour. Yeah.
All right.
Yeah, the room just kind of accidentally got opened early, about 20 minutes early.
Okay.
Yeah, but yeah, you can just relax for 5 minutes.
Yeah, yeah.
And then hopefully relax for the rest of the room, too.
Yeah, yeah.
There we go. That's been shared there, and we have the presentation up. There we go. And welcome everybody to Science Society. We're very, very glad you could join us. It's going to be an amazing talk, so hope you're all ready.
Katarina, have you been swimming in the ocean?
Yes, definitely. I love the water and swimming and everything about it. As soon as I'm by the ocean, I'm fine. Like, you know, could be the worst thing. That's why, I don't know, not living close to the ocean is when, in Porto or, whenever I'm really close to the ocean, stressful day, 2 minutes by the ocean, I'm okay. Like nothing can, must be
Yeah,
something really bad. Then,
it takes all the stress away. And it's rejuvenating.
oh, it is? Oh, perfect. Well, the intense sun, probably not.
Are you tanning in all the sun, Katerina?
I just am outside in the water. There's not like that I'm laying there and tanning on purpose. It's just, you wear sunscreen, but you still.
Right, golden brown. Yeah, just lovely. Yeah.
I used to like the
Um,
beach in New York. Like I never thought before I lived there that that was an option, but I actually really enjoy going to like Jacob Riis Beach or something and swimming.
Yeah, it's yeah, we have beach in Brooklyn, so Rockaway Beach. Yeah, it's pretty nice. I like it.
Hmm.
Just the pain to get there, like the traffic is mostly annoying. The
Yeah.
ferry hour from where we live, like Williamsburg has a ferry down,
Hmm.
but
Yeah, I used to just take the A train
yeah,
or
yeah,
something.
that's
Rockaway.
What's the temperature of your water?
Here it's relatively warm. So it's warmer than in Porto. In Porto, where I'm from Portugal, it's around 17 degrees Celsius, the water always year round. Maybe colder in the winter. So it's always really cold. Like it hurts the bones of your feet after a while. But I really like it when it's really hot and really cold water. So here it's warmer. It doesn't hurt your bones, but it's not warm. So, but I like it. It's like it gets too warm. I don't know. It's not refreshing. It doesn't feel like the ocean. I don't know. I don't like it. I'm weird.
I
I
get
was
it.
at Greece and stuff in the Mediterranean. I really didn't like it that much. It was weird. Like, what the heck? It's hot outside. You don't really cool down in the water.
Gulfside Florida is almost bathwater this time of year.
Hey Serena, how you doing?
Hello. Hi, everyone.
Hey, Serena.
Awesome, you're back in here tonight. Excellent to hear you.
Well, the topic was quite a draw.
Mm-hmm. Definitely.
Now, I've been so busy lately, but I'm trying to come back.
Glad you're here.
Very, very glad to hear you back, yeah.
Yeah, because I'm a little concerned about you and the gators and how close you let them get to you
Let's see my
after
new
you found out.
handbag. No, I think Marco, I haven't seen Marco in a while. They may have taken him.
Oh, that's sad.
I still keep a lookout.
I think we can slowly start. Yeah, with the introduction and everything, and then we'll go from there. So welcome everyone to the Science Society, and of course, a special thanks to Cliff, Dr. Cassidy, for coming here and sharing your time and your amazing research with us. Before we start, let me introduce you a little bit to the audience, and then Victoria will ask you a couple of interview questions, and then it's time for your amazing presentation because I had the chance to look at it. So, Dr. Clifford Cassidy is a scientist at the Royal Institute of Mental Health Research, which is affiliated with the University of Ottawa. And he received his bachelor in physiology and master in neuroscience and PhD in psychiatric research from the McGill University in Montreal, Canada. And then he did his postdoc on a fellowship in brain imaging at Columbia University in New York City. And he continued his neuroimaging work in schizophrenia and has expanded his research scope to the study of PTSD and other neuropsychiatric conditions. And he is currently also doing research with the military mental health unit, and he uses different and really interesting neuroimaging methods to understand brain mechanisms of different pathologies in the brain mostly. So thank you so much for being here. And yeah, as I said, Victoria will ask you a couple of questions and we'll take it from there. Thank you.
All right, thanks for having me.
Thank you. Thank you, Katarina. And yes, Clifford Science Society is so happy to welcome you. And it has been fascinating to read about the breadth of your research, and we can't wait to hear your discussion. So to carry us into your discussion tonight, I've just got a few questions to help us get to know you a bit and to carry us into your talk. So if you can reflect on your experience, maybe in childhood or any time in your life that you can recall when you first realized a strong connection or attraction to science or scientific exploration, then perhaps you can tell us about that.
I mean, yeah, I don't know that I had like a eureka moment or something. I think I was kind of an average child in terms of, earlier in my schooling. But then at some point in high school, I kind of just got really interested in science and math. And, I wasn't so sociable. I kind of felt like in a bit of a bubble. And I just got, it felt kind of reassuring, like a world that you could control or understand, like an objective world with clear rules. And so that was, yeah, kind of a comforting thing for me. And I just got really interested in it. And so it was kind of nice too, because then that gave me a kind of a springboard to get out of my town and explore the world. And so I went to McGill University in Montreal, which was on the other side of the country, when I was 18 years old, and started my studies there. And then that kind of got my life going too. Then I had a social life again. And so, I like the idea of science being also a kind of portal to meeting people in the community and being, engaging with other like-minded people. Yeah. So I didn't know at first if I wanted to do neuroscience. Like I, I was always interested in the brain. I remember even at a young age, being curious about that. But then started out in like the engineering stream for the first year of university, and then I decided to try neuroscience. I was always, curious about the brain and how our experiences can be, um, our reality is kind of shaped by whatever chemical reactions happening inside your brain. So I was interested in, psychiatric conditions that have such an change on your mood and your, sense of reality. And so I guess that's what got me interested in schizophrenia. I was also interested in substance use disorders. And yeah, that's how I started.
Thank you for that description. It's fascinating to hear also that you were so interested in the brain as a child, and then you continued that that search into what you were interested in. So can you then carry us from that point where you left off up to where you are today with your current research? Maybe
Yeah, so,
some stops along the way. Thank you.
right. Yeah, it's a good point that neuroscience was just the beginning of, finding your path. So that's still a very broad path. And I started out in doing animal research and basic neuroscience for my master's, which I liked, but I felt like I was also wanted to understand the human side of psychiatric conditions. For instance, for much of my training, I was studying schizophrenia, and I felt like I didn't actually know the kind of lived experience of people with the disorder. And so then for my PhD, I was based in a clinic for people with first episode psychosis, which I found really gratifying to actually work with the patients and to get to know them and to understand how the kind of illness interacted with their own personal selves. And so, but then I felt like I wanted to get a little bit back into the neuroscience side, because that situation was more like a clinical research kind of environment, which was very informative and educational. But so for my postdoc, I think I found a pretty good compromise where I was doing kind of human neuroscience work with learning about brain imaging, also mostly in schizophrenia. And yeah, so I kind of got back into more hardcore neuroscience, but could apply my understanding of the illness from, having worked with patients too. And then when I got my own independent position here in Ottawa, something happened where towards the end of my postdoc, we started working on this one method that I'm going to talk a lot about today, this neuromelanin-sensitive MRI. And so then I went from having a common thread of my research being a disorder, schizophrenia, to having the common thread being more the method that I was using with this neuromelanin imaging, and then applying that to many different disorders, including neurodegenerative disorders like Alzheimer's, but also psychiatric disorders. And that's been kind of interesting, because when you have expertise in a method, it kind of opens up a lot of collaborations because other people want to, maybe use it also, and you can help them. And so, yeah, that's been neat to collaborate with people around the world who are trying to apply this method in different contexts and different disorders and so forth.
Yeah, thank you. It's interesting that you had mentioned that science is a portal to meeting people and bringing us together into community, because that's really a goal with us here in Science Society, to bring science, love of science and interest in science to as many people as possible. So thank you for being part of our goal and doing that.
Yeah, I think a lot of scientists feel the same way, and it's great when you get a chance to share your results with people who are interested.
So interested. And you'll see, because so at this point you're welcome to deliver your talk, and then at the end we can have your Q&A if you'd like, or if you prefer the questions to drive your talk, your conversation, that's fine too. And we are here to moderate the room, and also sometimes people write questions in the room chat, which we can also share with you, so you don't need to worry about that at all. And I feel like I was going to answer one other thing that you said, but yeah, perhaps not, or you're here. I know where you are if I need to say
Okay,
it.
yeah.
So, all right, yeah, so yeah, we've got your talk pinned up at the top, so you can refer to that at any time. And so, yeah, thank you so much, and please enjoy your talk.
Okay, yeah. I mean, I'm happy if anyone wants to interrupt me as we go. I don't mind, and we can also have some extra questions at the end. I'm pretty flexible. So I'll start the talk, but I'll try to keep on track with telling the slide numbers, because don't want to get mixed up and lost. But if people, I'm sure I'll forget. So feel free to interrupt me if you get lost with where I'm at. So yeah, thanks again for inviting me, and I'm going to talk about this paper that I recently published called Association of Locus Coeruleus Integrity with Braak Stage and Neuropsychiatric Symptom Severity in Alzheimer's Disease. But I won't launch right into the paper. I'll give a fair bit of background, because kind of to give a perspective of where I'm coming from and how I got to this particular study and where this work is maybe leading. I'm kind of taking the perspective more about studying catecholamine neuromodulators, which a lot of my work has been focused on recently. So Alzheimer's disease was one application where those brain systems are affected, especially the norepinephrine, like the Locus Coeruleus norepinephrine system. But then we've also been looking at the dopamine system. So, those two systems are somewhat related, but certainly the neurotransmitters themselves are closely related, and I'll explain why we end up looking at the two of them sometimes with this type of imaging method we're using. So to start with the overview, so I'll talk for a while about the background of this imaging method we're using. So it's called neuromelanin-sensitive MRI, and so I'll explain a bit what neuromelanin is and the biology of that, and then how we use this imaging method that's related to neuromelanin, and then we will go into how some validation work, like how we kind of tried to establish this method as a real method to look either at neuromelanin or at catecholamine system function. So, which would be kind of cool, because usually if you want to understand those kind of brain systems, you have to do PET imaging, which is a great method, but has a lot of limitations, and it's not always possible to use it in a lot of contexts. So we were excited to try to do something with MRI that could tell some kind of similar information. And then once we've gone through all that, I'll move on to the actual results of the study that we want to talk about today, about using the method in Alzheimer's disease to examine degeneration of the locus coeruleus in that condition. So I'll start with the background. So why are we talking about catecholamines? Well, I mean, I guess I'll have to give a little background here. So catecholamines, usually we think of dopamine and norepinephrine, and So they're very closely related chemically. And we care about them because, you know, these systems are such important neuromodulators that are involved in so many brain functions. In the context of what we're talking about today, it's really important to know that these systems are vulnerable to neurodegeneration, and we'll get into what reasons that may be, why that may be the case. A lot of psychiatrists target these systems, like the dopamine system, norepinephrine system. And yeah, they're involved in really key brain functions like cognition, memory, arousal, especially the locus coeruleus and autonomic function. And as I said, that it's been hard to, you know, in many contexts to directly measure the function of these systems in humans in vivo. So often people are using PET imaging, but I'll introduce this neuromelanin-sensitive MRI that we've been studying so much, and that is a way that targets both the dopamine system and the norepinephrine system, and allows us to assay the function of and the integrity of those systems. So a little bit of background about neuromelanin and catecholamines. So now I'm on slide 5. So here we see the nerve terminal of we'll talk about the dopamine system for now. The same more or less should apply for the norepinephrine system. So if we imagine the dopamine system nerve terminal, there's a vesicle that has dopamine in it, and then the dopamine is, you know, the vesicle releases it into the synaptic cleft, and then the transporters take the dopamine back up into the cytosol of the nerve terminal, then it goes back in the vesicle. So there's kind of cycling around. But the part in that cycle that matters for us right now is when the dopamine is free floating in the cytosol. So when it's in there, it's kind of a risky place for it to be, because dopamine and catecholamines in general are dangerous species. They're put oxidative stress on the cell, and they shouldn't be floating around at high concentration for a long time. So the cell does a lot of work to mop them up. They get sucked back up into the vesicles, but if there's a lot of them floating around, some of them will get turned into neuromelanin. So that isn't kind of an oxidative process. And then, as you see from the slide before, slide 4, neuromelanin is very closely chemically related to dopamine and norepinephrine. So that neuromelanin, it accumulates in these granules, and then those granules go into the cell body, and they just build up over the lifespan, and they never go away. So it's kind of like, maybe it's kind of like garbage, but it serves a function also. And the only way they'll ever go away is if the cells die, and then the neuromelanin can be cleared by microglia. So basically, as people age, their catecholamine neuron cell bodies accumulate more and more of these neuromelanin granules. So that's what you can see from this scatter plot here, that people who died at different ages, when they tested the amount of neuromelanin in their neurons, that increased gradually over the lifespan. So that's by our colleagues in Italy who helped us with some of the work we're going to show later. So the only way, and you see people with Parkinson's disease who've had loss of death of some of their dopamine neurons, then you see the neuromelanin levels are much lower. That's on the next slide, 6. So that's interesting, and it kind of shows how neuromelanin could be a proxy of integrity of cell bodies for catecholamine systems. But then for us, when we started this work, especially, we were thinking more about psychiatry and conditions that aren't having neurodegeneration necessarily. But we thought that even in the absence of neurodegeneration, the amount of neuromelanin could be telling us something. So there was work that showed that if you flood cells with really high levels of catecholamines, they will produce more neuromelanin. That was just work in the dish and in vitro. So we, because we were studying schizophrenia, where it's well known that there's increased turnover of dopamine and increased presynaptic dopamine levels, we had a strong hypothesis that could lead to increased accumulation of neuromelanin in the cell bodies. And there was a couple of small studies that suggested that might be the case. So based on this, neuromelanin levels could vary based as a function of dopamine tone kind of over the long term, because, it's something that would change only very slowly, right? That the neuromelanin is very stable, but gradually increasing and accumulating. So if there's a sustained increase in tone, that might lead to a gradual increased level of neuromelanin. And the other way that the neuromelanin levels could vary was if this dopamine cells die. So this idea of imaging neuromelanin started about 15 years ago by a group in Japan. And so they noticed that there's a special type of MRI sequence that could show a high contrast in the same area of the brain where neuromelanin is present. I forgot to say that neuromelanin is a black pigment, and that's what gives the name of the substantia nigra. It's nigra because it has this pigment in it that you can see with the naked eye. And also the locus coeruleus, it has the same pigment, but it's more diffuse, so it looks blue instead of black. That's what coeruleus means, blue.
Quick question.
Yeah.
Is the neuromelanin vesicles, are they distributed in any particular region within the cell, or do they just sort of...
They're just in the cell body. So that's why, it could be that there's some getting transported in the axons or dendrites, but the vast majority would be in the cell body. And there's probably low levels of neuromelanin throughout the brain, but it's very minimal. And then the high concentration is in only those two places in the brain. And that's why you can see the pigment. So yeah, these early work showed that both of those regions, the substantia nigra and the locus coeruleus, had a high contrast that was in the exact location of those two structures where the neuromelanin was present. So they called it neuromelanin-MRI, and there's still debate the extent to which neuromelanin contributes to the signal. So that would be a discussion we could have. There's some camps who debate whether other factors are more contributing more to the signal. I mean, it's the case in all MRI measures that we want to use the signal we're measuring as a proxy of some brain process, but it's probably impacted by many things. Anyways, so we could talk about the physics of it later. But so most of the initial applications of this imaging were in Parkinson's disease, and there's been many, many studies always showing a profound effect that people with Parkinson's disease have less of this bright signal in the substantia nigra, which presumably is related to the loss of their dopamine neurons. And so, yeah, at this point, if I go to slide 12, just quickly showing a meta-analysis that all the studies show a strong effect, and it's very consistent that it's a good method, for instance, in Parkinson's disease, to as a kind of a biomarker of the illness and the process that's really a core process of the illness, the death of the dopamine neurons. So then on slide 13, I have shown also in Alzheimer's disease that other groups have shown, similar to the study I'm going to present, that there's loss of the signal there, likely related to degeneration of the locus coeruleus, which is known to happen in Alzheimer's disease. So, that's in the context of today's talk, almost that's all I had to say. Like this is a measure of neurodegeneration. But I feel like I wanted to kind of give a bit of a broader background about how we could also use the measure to look at the function of these systems, even in non-neurodegenerative conditions, because I think it kind of informs how we interpret the results we have in Alzheimer's. And anyways, so I'm going to also talk about the work we've done in non-neurodegenerative conditions and how we've looked at the signal there. And it presents, that's a great asset that we can use it in different contexts, but it has a bit of a caveat also that to keep in mind when you use it in neurodegenerative conditions, it may not be just a marker of neurodegeneration. Like you can't necessarily separate the impact of neurodegeneration on the signal from the impact of other factors that impact neuromelanin levels. Or other factors that just impact the signal one way or another, even if it's not related to neuromelanin. So we're going to talk about that. So how we've used, now we're back to the overview slide, neuromelanin-sensitive MRI as a measure of catecholamine system function. So I'll talk about it in terms of the dopamine system for this part of the talk. I'll kind of touch on this topic at the end, how we've considered the norepinephrine system also. But anyways, for now, just the dopamine system. So the first question we addressed was whether, in fact, this is neuromelanin-sensitive MRI. It's called that, but is it, in fact, true to the name? So we worked on these post-mortem experiment where we had small half midbrain pieces from post-mortem tissue, and we scanned these using the same sequence we use, the neuromelanin-MRI sequence, and then we dissected them on a grid. So we made this little 3D printed dish with a grid insert, and then that way we could register the images from the MRI with the chemical measures from the dissections. And so our objective was to see if the grid sections where there was higher signal also had higher neuromelanin content. So on the far right on slide 18, you see a representative specimen, and on the y-axis is the concentration of neuromelanin that was measured using spectrophotometry from the dissected sample. On the x-axis, you see the signal that was measured with this neuromelanin sequence. And you see that the grid sections that had higher neuromelanin content had higher signal, as we expected. The scatter on the left is just combining all the 7 specimens together. And so you see that, indeed, that the neuromelanin concentration is correlated to the neuromelanin signal. So it argues in favor that neuromelanin is impacting the signal. So then moving on from that question to the bigger question of whether this is telling us something about the function of catecholamine systems even when there's no degeneration happening. So for that work, obviously, we're working in vivo. So I'm going to talk about our methods to look at the substantia nigra in living people. So a lot of the early work in Parkinson's disease had simply done a very simple analysis approach where you just work on the raw image and draw a circle around the bright part, which you assume is the substantia nigra, and you look at the contrast there relative to the signal there relative to the signal in a nearby reference region where there's no neuromelanin, which is, I'm not showing here, but it's the white matter tract right beside the substantia nigra, the crus cerebri. So you look at the contrast between those two things. But the limitation of that approach is, well, there's a couple, but the main one is that you're not able to look at subregions within the substantia nigra. And it may be important to do so because even though the substantia nigra or the locus coeruleus also, even though they're very small structures, that doesn't necessarily mean they're homogeneous. And for instance, we know the dopamine system has different branches to it. There's a limbic dopamine system, a motor dopamine system, and a cognitive one, or associative. And those have some, anatomical specificity. So we thought it was important to be able to break up this structure in greater anatomical detail. So we did what we call voxel-wise analysis, where we could look at all the different individual voxels within the substantia nigra. So to do that, we had to go to standardized space. So we registered everybody's brain. The neuromelanin image, as you can see in the slide 20, there's a red band there. That's the neuromelanin image overlaid on a standard anatomical image. So it's not a full brain scan. It just is a smaller field of view that covers the midbrain and the pons, which is where the nigra and the locus coeruleus are. So we brought that neuromelanin scan into standardized space, and then we mask out where the substantia nigra is. You see the red in the intensity normalization. It has a red mask where the reference region is, where there's no neuromelanin. And then we create this kind of standardized space template of the substantia nigra here. on the next slide, 21. So you can see that yellow mask is the substantia nigra, and the pink mask nearby is the reference region. And then we can look at every one of those little squares inside of it and do an analysis on each square individually. So one of the first things I'll show on slide 22, one of the most basic things we understand, if neuromelanin is really related to this neuromelanin imaging, we would have a strong hypothesis that the signal should increase over the lifespan, because we saw that with the postmortem tissue, that the neuromelanin levels increase over the lifespan. And we found that indeed that's the case, that if you see here, this red mask over the nigra is the red voxels where we saw significant increased signal over time. What was interesting, we saw if we span the whole lifespan like we are here, also there's a part of the nigra that starts to go down in signal with age, and that's on the periphery of the structure, which would suggest that the structure is shrinking a little bit. So the middle is getting brighter. It's maybe shrinking in size a little bit as people age, maybe due to some degeneration as part of healthy aging. So if we do this as a scatter plot, again, on slide 23, we see that in this one large study of people from age 20 to 85 or 86, we see that the signal is increasing in those red voxels. In another smaller study just on younger people, we see that it's increasing even more steeply at younger ages. And there's no voxels there that are decreasing with age. So that seems to be happening only in later life, that maybe the nigra is shrinking like that. So this is just a small taste of another study that's going on now in healthy children and adults. So especially we're interested in the children to understand how the dopamine system's developing in healthy kids, because it's something that's never really been able to be studied directly in children. So a lot of the knowledge we have about the dopamine system's development is from animal research. You can't really do PET imaging in youth. So we're pretty excited about this study, and we might be applying the method also to kids with ADHD, for instance. Hopefully that study will start soon. So that's a sidetrack, but the other thing we wanted to do is maybe the most important validation that the method is relevant as a measure of function of catecholamine systems was to correlate the signal in the substantia nigra to kind of like a gold standard measure of dopamine function, which is from PET imaging. So the group that I was working with when we did this work at Columbia were experts in PET imaging, especially dopamine PET imaging, and schizophrenia as well. And so we were using a measure, using a tracer. So now we're on to slide 25. A dopamine radio tracer called raclopride that binds the dopamine D2 receptor. And if you scan someone following injection with that tracer before, and then you scan them again after injection with amphetamine, which causes great amount of dopamine release, you can see the displacement of the tracer by dopamine, or by amphetamine, sorry, that would give kind of a proxy measure, or yeah, by the dopamine that was, released from the amphetamine. So that gives a proxy measure of dopamine release capacity. So it's sort of like how much presynaptic stores of dopamine were there. So we had a hypothesis that people who tend to have higher dopamine release capacity, maybe if that was sustained like a trait-like way over their lifespan, that might lead to increased neuromelanin accumulation, and we would see brighter signal in the substantia nigra in those people. So that's what we found. We found a cluster, these green voxels on slide 25, that had higher signal in the people with higher dopamine release capacity. So that kind of suggested this measure could be capturing something about dopamine system function, which is pretty exciting because then it can be used outside of just neurodegenerative context. So the next step from that was to consider how the signal might be affected in a condition where we know there is a change in dopamine function, in presynaptic dopamine function, which was psychosis. So we know that people with psychosis have elevated dopamine system function, elevated dopamine synthesis, elevated dopamine release. And so we tested that in our study with neuromelanin-MRI as well. And we found that patients with schizophrenia and also patients at risk for psychosis, both of them had increased neuromelanin-MRI signal that was correlated to the severity of their psychotic symptoms, or subthreshold psychotic symptoms in the case of people at risk. And we found that both of those clusters within the substantia nigra, where there was a relationship to psychosis, were kind of clustered on the ventral substantia nigra, which is the part of it that's supposedly connected to the associative striatum, which is the part of the striatum where there's elevated dopamine release in schizophrenia. So that was pretty cool. We have our colleagues are working now on using this as a marker of predicting treatment response in schizophrenia. They've shown that hasn't been published, but that this is predicting treatment response, which is exciting because a lot of patients with schizophrenia don't respond to the first-line treatments, and then they have to use a special drug called clozapine, which can be challenging to use and isn't offered always until late in the illness process. So if you could know who needs it sooner, that would be great, especially with a method like this imaging, which is pretty practical to administer in a clinical setting even. So that summarizes kind of where we were at with using the method. So we had a brief introduction to how it could be used in the context of neurodegenerative conditions, but it can also be used in the absence of neurodegeneration, since the signal correlates to neuromelanin concentration, increases with age, correlates to dopamine release capacity, and is elevated in psychosis, where we know there's elevated dopamine function. So, I think that's all worthwhile to keep in mind when we're looking at using it in Alzheimer's disease. So we know that it should capture degeneration of the locus coeruleus there, but it may also be sensitive to other things that are happening in Alzheimer's disease. So I'm going to start the talk about the work we've done in Alzheimer's now. So now I'm on to slide 30. So now enough about dopamine, we can talk about the locus coeruleus norepinephrine system. So presumably things are similar in terms of imaging this system as they were imaging the dopamine system with neuromelanin-sensitive MRI. But to backtrack to talk about the biology of the locus coeruleus and its impact in Alzheimer's. So locus coeruleus is a very small structure. It's kind of rod-shaped, and it projects very widely throughout the brain, and also down to the spinal cord. It's this norepinephrine neuromodulator is very important for many functions, including arousal and memory, and autonomic system function, also sensory function, motor function. Like it has a lot of roles. So in Alzheimer's disease, there's a few reasons why this is a particular target of interest. So the locus coeruleus is kind of unique in Alzheimer's because it accumulates this tau protein before any other brain region. So why that is, there's a lot of work to be done to understand that better, but it's exciting to think that this could, this structure could be one of the early players that kind of triggers the cascade of events that leads to Alzheimer's. So we'll talk a little bit more about that. Later in the illness, the LC degenerates, and then as it's degenerating, there's kind of compensatory changes, so that even though the cells are dying, the norepinephrine cells, the remaining cells, they show some aspects of kind of hyper functioning. So maybe they're kind of even potentially overcompensating sometimes and causing other problems. So I'll just briefly touch on a few of those things. So in terms of the early tau accumulation, it was, there was a kind of idea about the development of Alzheimer's into different stages, which they call Braak stages. So that's based on the spread of tau throughout the brain, this tau protein. And it was after the Braak developed the stages. They had stage 1, 2, 3, 4, 5, 6. based on the spread of tau. Shortly after, they realized they needed to have also a stage 0, which is when tau is only present in the locus coeruleus. And that can be seen even in the 20s and 30s. So when people are between, second and third decade of life, there's already some tau in some people in the locus coeruleus. And that tau, accumulates more as the illness spreads. How important it is, we don't know, but, people theorize that could be a source of tau that, spreads it throughout the whole cortex eventually, because certainly the locus coeruleus projects throughout the whole cortex. So then the question about why the locus coeruleus is vulnerable to degeneration, there's different ideas about it. There's perhaps many reasons. The locus coeruleus is close to the fourth ventricle and to blood vessels, which exposes it to cytokines and toxins. I think another special thing about these cells is they have these huge axonal arbors, and very narrow axons, I think, too, maybe. So it's kind of a difficult, it's a lot of work for the cell to maintain that huge axonal arbor. And the other thing, of course, is that these neurotransmitters they're using, the catecholamines and norepinephrine, for instance, causes oxidative stress on the cell, and that leads to some kind of cascades that can cause damage to cells. And neuromelanin is part of that process of trying to protect against oxidative stress. But then, even though it's protective, in certain situations it can kind of get reversed and become harmful. If neuromelanin, for instance, is released from those granules it's supposed to be protected in. So, it's sort of like mopping up the bad stuff, but then if there's a situation where it gets released, then that bad stuff is going to cause damage. And then just briefly to touch on how the locus coeruleus norepinephrine system may change in response to the damage or the death of cells. There's evidence of a number of different changes. For instance, elevated synthesis of norepinephrine in the remaining cells, elevated receptor expression of norepinephrine receptors, and increased sprouting of nerve terminals also. So there's a lot of, it's kind of hard to keep track of all these changes. So there's the tau accumulates in the locus coeruleus, then eventually the cells start dying, then the remaining cells have these other changes. So a lot of different processes are happening, and it could be hard to disentangle them all. So in terms of our study, one aspect we were excited to understand better was the role of the norepinephrine system in terms of neuropsychiatric symptoms of Alzheimer's. So, of course, Alzheimer's presents with cognitive deficits and memory deficits, and there's most likely that locus coeruleus norepinephrine system is important for those, but it also, in many cases, people with Alzheimer's have other types of behavioral symptoms. For instance, apathy, mood disorders, mood changes, and even psychotic symptoms like reality distortion, paranoia, impulse control problems. So there's been a fair amount of research linking those symptoms to these changes in the norepinephrine system. So we thought this was an opportunity to understand that better as well by using this imaging method. And yeah, some of these types of neuropsychiatric symptoms have been able to be treated with drugs that lower norepinephrine system function. So even though the norepinephrine system's dying in Alzheimer's, it seems a bit counterintuitive, but we know that it's also hyperactive. So drugs that are dampening the system, despite it being damaged, can actually help in some cases. So now we'll talk a little bit more about how we use this neuromelanin-MRI to look at the locus coeruleus. So similar to the substantia nigra, we see the high contrast in the structure. So I'm on slide 35 now. And here, this kind of computer-generated image shows results from a cell counting study that's kind of like the ground truth of what the locus coeruleus looks like. And you see it's kind of like a rod-shaped structure. So in a cross-section, it's very small. So to image it, we had to, it's challenging to image it and to analyze the images when the structure you're looking at is very small. But we developed a special method to do that. As you can see, also the locus coeruleus, you can see it in the cut specimen as a kind of dark blob. So on slide 36, we talk about the human locus coeruleus imaging we're doing. So you can see again that the bright signal there by the fourth ventricle, where the locus coeruleus is, and then in Alzheimer's disease, the signal is greatly reduced in many cases. So for our study here, our objective was to determine the extent of locus coeruleus loss in Alzheimer's disease, like where and when is the signal lost in vivo, because I said it's lost after the tau accumulation, but when exactly in the course of the illness, and what part of the structure. When I said where, like is all the structure affected, all the locus coeruleus, or only some parts of it more affected than others? And in general, it was also a good opportunity for me using this method, developing a new method to look at the locus coeruleus with neuromelanin imaging. Before we tried to apply it to more novel situations like in psychiatry, for instance, we wanted to see if it worked in Alzheimer's disease, where we know we have a very strong hypothesis of what to expect to see, and other groups have already done some work in this area. So it kind of served to help us develop our method a bit too. So, and the other thing we were especially interested in, like I said, to understand this relationship between the locus coeruleus signal and these neuropsychiatric symptoms. And we thought that, they would be positively related. So either people with high or preserved locus coeruleus signal would have worse neuropsychiatric symptoms. So in this case, I was collaborating with a group at McGill University, so I was very lucky to work with them. So at the McGill University Research Center for Studying in Aging, they have an ongoing, very large study of people with Alzheimer's disease, but also across the whole spectrum of Alzheimer's, from healthy aging to people with mild cognitive impairment to Alzheimer's at different severity, although they don't have a lot of people with very severe Alzheimer's. But their data set has a lot of great data in it. They have a lot of clinical characterization of these people, different neuropsychiatric or neuropsychological measures, and a lot of imaging measures as well. So everybody in this data set has not only MRI with neuromelanin-MRI and other types, but also tau and amyloid PET imaging. So everybody has 2 PET scans with the different tracers. And then they have other markers as well, but we won't talk too much about that. So just looking a bit more at this sample on slide 41. So it's a large sample. There's almost 200 people. We had 28 people with a clinical diagnosis of Alzheimer's, 44 with mild cognitive impairment. But in the end, there's a movement in Alzheimer's research not to use the clinical diagnosis anymore, because now we understand the illness well enough to know really what the biological definition of the illness is. So for most of the study, we broke people based on whether or not they had tau in their brain. So we divided people into tau positive cases. So there's a few people who were considered cognitively normal, healthy older adults, but they did have tau in their brain, so it hadn't manifested into any symptoms yet. And then the proportion was much higher in people with mild cognitive impairment, and almost all the people with Alzheimer's were tau positive at this threshold. So that's how we looked at these people. So now going to slide 42. So a lot of the work I was doing was developing a way to measure this signal in this very small structure from these neuromelanin images. And other groups had done similar work, but I wanted to try to find a way to do it pretty much automated. We double check and look over the results, but it's an automated pipeline. And it can kind of capture the bright locus coeruleus signal, but in a flexible way so that we don't, miss it some of the time. when it's so small, if you use the usual approach in MRI, where you take a mask in standardized space and just apply that mask on an image, if it's a very precise mask, it may miss a very small structure like this. If it's inaccurate by 1 voxel, you could miss the whole structure. So we came up with a method where we can look at the images on the raw, unprocessed, undistorted image. So we kind of create this search space in the standardized space, then we carry that search space to the native space, and then within the search space we find a bright cluster of voxels that we call the locus coeruleus. And we do that across the whole extent of the locus coeruleus because even though it's small, it's rod-shaped, so we can divide it along the rostrocaudal axis. So we can get not just a single measure of the locus coeruleus, but 5 measures. So we have kind of the top, middle, bottom. So we can have some sense of if different parts of the structure are more implicated in certain aspects, like in severity of Alzheimer's or relationships to neuropsychiatric symptoms. So it's an automated pipeline. We made a couple of corrections manually when we found it wasn't working right. Sometimes there's artifacts on the images that can affect it. And we also did manual segmentation, which was quite laborious, but it gave us an opportunity to compare our automated method. We found that they had very good agreement. So it's a much less labor intensive, potentially more practical, and could be used in clinical settings, and more objective also than a manual method of finding this structure. So moving on to the results. So now I'm on slide 44. So the first question, we wanted to just think about how the locus coeruleus signal was affected in Alzheimer's. So we looked at people who were considered tau positive. That's our definition of Alzheimer's. And we looked at tau not in the whole brain, but in this particular region of interest, which on slide 44 is this red region of interest in the temporal cortex, which is where tau starts spreading in Alzheimer's, and especially the entorhinal cortex. So if we define people who had a certain level of tau in there as tau positive, we compare those tau positive people to tau negative people, we found that there is a great reduction in the locus coeruleus signal in the tau positive people. That's if we just look at the whole locus coeruleus, average the whole structure together. But then if we break it into these pieces, so we have 5 pieces on the left, 5 on the right, and we have this kind of color code. So this is kind of a schematic of the locus coeruleus on slide 45. We see that the darker blue is the places where there's greater loss of signal. So we found that the very middle of the locus coeruleus and the section just below it had significant signal loss. The section above it was close, so there's probably loss happening there. The ends of it, the rostral and caudal ends, didn't have significant loss. That's kind of a noisier place to measure, so I'm not surprised it didn't come out significant. So it was interesting if we compare, because here we saw the middle of the locus coeruleus had the greatest signal loss. If we look at data from cell counting studies on slide 46, so previous work where they looked at the amount of cells that die in Alzheimer's from locus coeruleus tissue, they found that it was in the middle of the locus coeruleus where there's the greatest loss. So here the x-axis is the cell count, and the y-axis is the distance from the top to the bottom of the LC. And the two curves, the curve on the right is healthy people who died without Alzheimer's, and the curve on the left is people with Alzheimer's. You see that this kind of blue arrow is showing the amount of loss of neurons, and that loss is happening in the middle of the LC, which is where we saw the greatest signal loss. So it kind of confirms that our method is capturing something that makes biological sense in terms of what we know about Alzheimer's. And then the next question is, when in the course of the illness is this loss of signal happening? So, and presumably the loss of the locus coeruleus cells. So if you see on slide 47, the subplot F, shows on the x-axis the Braak stage, which is kind of the progress of Alzheimer's from stage 1 to 6. And the y-axis is this signal in that red encircled part of the locus coeruleus where it's most affected. So it looks like there's kind of a steady loss of signal as people progress in Alzheimer's disease. But one thing that, I was intrigued about, and I didn't talk about too much, a little bit in the paper, but, it's part of the reason I did this whole setup about how, this neuromelanin imaging is probably not just measuring degeneration. So if you look at this subplot F, it looks a bit like the signal goes down sharply from Braak stage 0 to Braak stage 1. And then it kind of looks like it's going up from stage 1 to 3, and then it drops sharply from 3 to 6. So why would it have this little rebound there? And, maybe it's just noise in the data. It was statistically significant if we put, a cubic fit onto that. But it could be that, it may be related to some of those compensatory processes that are happening. So when the cells start dying, then they start getting hyperactive, could lead to increased neuromelanin or other changes in the cells that is increasing the signal in a kind of transient way in the intermediate stages of Alzheimer's. And then it starts, then maybe the cell loss overwhelms that, and then it just goes down precipitously from there on. Anyway, but if you just fit a simple linear fit, you see a gradual decline of the signal as people progress through the illness. And then on slide 48, you see that similar to that, if you look at not Braak stage, but severity in terms of cognitive impairment or overall dementia severity, in both cases, people with worse symptoms or severity have lower signal in the locus coeruleus in that mid-caudal section that's encircled in red there. So all that data kind of fell together pretty nicely to confirm that, the signal's lost as we expect in Alzheimer's disease. It seems to be lost gradually over time, especially in the middle of the locus coeruleus, and gets more lost as people get sicker. I'm going to skip that. So slide 50, then we talk about how this signal in the locus coeruleus correlates to severity of neuropsychiatric symptoms in Alzheimer's. So we had a hypothesis, even though it's kind of counterintuitive, the signal is decreasing in Alzheimer's. We thought that it would be positively related to worse neuropsychiatric symptoms. And that's what we found. So that's consistent with other work too, using different types of measures of the Locus Coeruleus norepinephrine system, that people with higher signal people with higher norepinephrine system function have worse neuropsychiatric symptoms. So in the scatter plot here, that's what we see, that the y-axis, let's go to slide 51 and we can see that more easily. Sorry, slide 50 is showing that is independent of other changes that are happening in the brain in people with Alzheimer's. So that's in this table here on slide 50. It's showing that, it's not, the trouble with studying Alzheimer's is everything is changing at the same time. And if you just look at one measure, you don't know if that measure is really, one biological measure is related to a clinical problem, because probably the people who have worse or more advanced degeneration or change in that biological measure have worse on all the other biological measures also. So it's interesting to see if the biological measure is independently related to a clinical measure, independent of other biological changes in the brain. And that's what we found, that the LC signal was predicting severity of neuropsychiatric symptoms independent of the accumulation of tau, independent of the accumulation of amyloid, and independent of loss of gray matter in the cortex. So that's putting all that stuff together, we were able to predict the severity of neuropsychiatric symptoms pretty well. So back onto slide 51, if we look at just a scatter plot of the LC signal and relating that, again, the LC signal in the mid-caudal section, relating that to the severity of neuropsychiatric symptoms on the y-axis, we saw that they're positively related. And it was especially true if we, at first we were just looking at overall severity of neuropsychiatric symptoms, combining all the types together. But if we break them down, we saw the subdomain that was really most affected was what they call impulse discontrol. So that's like aggressive and agitated behavior. So people with those kind of symptoms had relatively brighter signal in the locus coeruleus. So it could suggest that, if your locus coeruleus is preserved, that's a good thing. It might, protect you against some cognitive decline, might mean you're not as advanced in Alzheimer's. But it can also be a bad thing because it might mean you're more at risk of neuropsychiatric symptoms. So if you have other symptoms, other changes in your brain, other aspects of neurodegeneration happening in Alzheimer's, but your locus coeruleus is preserved, maybe there's some kind of imbalance where it can lead to neuropsychiatric symptoms. On the other hand, it could be something about hyperactivity of the locus coeruleus that's being captured here, and that hyperactivity is what's driving the symptoms. So either it's preservation or it's hyperactivity. The hyperactivity interpretations may be a bit more controversial because that's assuming that that hyperactivity could lead to brighter signal. So that's my take on that finding. I thought I'd just mention this briefly on slide 52. We also had a quick look at the substantia nigra in terms of neuropsychiatric symptoms. This wasn't in the paper, but we always get, when you collect these images, you can always look at both structures. And certainly the dopamine system is relevant for different types of neuropsychiatric symptoms. And we found that if we included that also into the model with all the other brain predictors, both the substantia nigra and the locus coeruleus were independently predicting severity of neuropsychiatric symptoms. And the substantia nigra was more related to what they call emotional dysregulation, which is more like depression and anxiety symptoms. So when you had lower signal there, worse symptoms. So I'm going to summarize the findings of this paper. So we found the locus coeruleus is gradually lost in Alzheimer's disease, presumably due to degeneration. The extent of the signal loss is correlated with Alzheimer's disease severity, including Braak stage. But the preservation of the LC in Alzheimer's disease can also be harmful because we found that the LC signal was positively correlated to neuropsychiatric symptoms, especially impulse discontrol. So in general, it suggests that this MRI can interrogate the role of the catecholamine systems in human studies of understanding mechanisms of Alzheimer's disease pathophysiology. And maybe it could have a promise in more of a clinical context as a biomarker of risk for neuropsychiatric symptoms or even risk for Alzheimer's. So I guess I had a couple more slides about how we've used the method in PTSD to look at the locus coeruleus in a non-neurodegenerative condition. I guess I could go over that. It's only a couple of slides. So here, we were kind of curious. So we, in our previous work looking at the dopamine system, we had looked at it in Parkinson's disease as a measure of, neurodegeneration of the substantia nigra. We looked at it in schizophrenia in a context where there's not neurodegeneration, but where there would likely be hyperactivity of the dopamine system. And so we wanted to see, in that case, how the method could be used in neurodegeneration or in non-neurodegeneration, like psychiatric conditions. So similarly in PTSD, we thought that could be kind of like how we looked at the dopamine system in schizophrenia, but the locus coeruleus could be similarly relevant in PTSD because the locus coeruleus is really important in regulating arousal. And one of the most important symptoms of PTSD in many people is hyperarousal, which is like kind of being always on alert, even long after any real threat has passed. So there's evidence that the locus coeruleus can be hyperactive in PTSD. And other studies have used this locus coeruleus imaging in context of anxiety or autonomic disorders, or showing that there's a relationship there. So we wanted to see if indeed that the locus coeruleus signal was higher in people with PTSD and if it was related to the severity of their hyperarousal symptoms. So we did this in a population of military veterans with PTSD. So we had here 32 Canadian Armed Forces veterans who had served in Afghanistan. We had 19 match controls. Everybody did the neuromelanin session, and then we did clinical assessment for PTSD symptoms and depression. We did the same method to look at the locus coeruleus signal. Because of the way the images were acquired, we couldn't break it into 5 pieces, only 3 pieces here. And we found in this case that it was in the caudal LC, the bottom of it, where we saw a positive relationship. That brighter signal there was related to worse hyperarousal symptoms. In this case, we controlled for depression severity also because there's been evidence that people with depression have lower LC signal using this method. So, and depression is often comorbid in PTSD. Also, if we looked at just the group effect, we found that people with PTSD had significantly higher, quite a bit higher signal in some cases compared to healthy controls. So it suggests that, the locus coeruleus may be hyperactive in PTSD and may be related to some of these symptoms patients have. So on to the final summary. Neuromelanin-MRI can be used to assay integrity of catecholamine nuclei in neurodegenerative illness, so like the substantia nigra in Parkinson's, locus coeruleus in Alzheimer's. It also appears to measure aspects of catecholamine function in the absence of neurodegeneration. So we looked at the substantia nigra in psychotic illness, the locus coeruleus in PTSD. And, the method can be used, has a lot of promise to be used in mechanistic studies to understand better, human brain function, and also as a clinical biomarker. So we have been working with the company to try to bring this to the clinic to get approval to use it in different conditions as a marker of potentially, ultimately as a marker of illness or that could predict diagnosis or treatment response. So that's it. So I have some acknowledgments for my crew, but happy to take any questions or discuss anything.
Thank you so much, Cliff, for this, amazing presentation that you went over through so much interesting data. It's very impressive work. Thank you so much.
Thanks.
And yeah, when I read this, like the PTSD data is really interesting too. That the hyperexcitability marker seems to be really significant and quite important. Did you have like how soon when PTSD? So usually we say PTSD starts around like clinically around 1 year, I think, after trauma, if it still persists. Like you have maybe patients before. Are you looking into patients to diagnose it earlier on and maybe prevent it, planning on doing that maybe?
Yeah, that's an interesting thought. I mean, if this signal is caused the way we think by neuromelanin accumulation, then it may take some time for enough change to happen to be detectable. But we don't know. We don't know how much time that might be. So it could be that it might be detectable before symptoms. That's a good thought. Yeah, like the people in our study, Canada left Afghanistan. I don't know how long, over 10 years ago, maybe, or close to 10 years ago. So they've all been, living with PTSD for many years. These people, we don't know how long this has been present. But that would be a really interesting question. When does it show up and how long does it persist? Like, is it there forever, for your whole life, even if you get better? Like, that's based on if it's really due to neuromelanin, you'd expect it's sort of like, kind of like a scar or something that, is just a permanent record, even if you get better, because the accumulations happen and it's still there.
Yeah, it's interesting because we see especially with PTSD, even if you do a lot of treatment, you have the spontaneous reoccurrence of PTSD or this traumatic memories. Even, I can.
Mm-hmm.
It's very common if you're stressed out again, but there's
Mm-hmm.
spontaneous recovery, even if you did a lot of extinction training.
Yeah.
So yeah, and then it would be also interesting to see data before they got PTSD, if there's some kind of already
Mm-hmm.
position, because that would be amazing to screen for example, for soldiers.
Mm-hmm.
If you don't send them, if they already,
Right.
have kind of a hyperactive, a little bit more active.
Yeah, that would be so cool. I mean, it'd be obviously challenging study to do, but you never know. Maybe, yeah. I mean, hopefully not, because that would mean there's people being exposed to war again or something. But if you could get people before a likely exposure to traumatic event, you could do that kind of a study.
Well, another group that would be interesting would be police officers and
Mm-hmm.
like firemen have a very
Yeah.
percentage of PTSD
Mm-hmm. Yeah, we've
in
thought
when
about that.
they start to become firemen, and then
Mm-hmm. Yeah, that would be, a long-term study, but definitely one that would be cool to do. Yeah, we're doing more work on this to understand it better, like doing a larger study and relating it to um, different things, like we're doing fear conditioning in PTSD. So um, through that, you get a kind of physiological measure of arousal, like skin conductance response, and different functional changes in the brain while they're learning, like fear learning. So yeah, we're hoping to dig into it better to see what's going on.
yeah. So the only interesting thing that would maybe speak at least partially against it being completely predisposition is that um, the high occurrence of PTSD in firemen. Because I did a language data study, a while ago, and compared this different police, firemen, abuse in childhood, all of these different groups.
Mm-hmm.
The thing is, in the language data, one of the highest indicators were use of pronouns. Like if you use I a lot, it's known for a while, then you're like more likely to be depressed and have all these
Mm-hmm.
PTSD and stuff. And then if you use we and stuff a lot, then you're kind of a little bit protected. Even in Holocaust survivors and so on, we kind of saw that. So then I talk with soldiers, and they go through this training where they kind of erase I. Like during that training,
Okay.
you're never supposed to say me. You're always, or I. You're always
Mm-hmm.
us. Like you're always supposed to see yourself not as yourself anymore, as a group.
Mm-hmm.
And I kind of thought that it might have this protective that firemen don't go through, and that's why PTSD is way higher in firemen. So it would be cool to do that study and then also collect language data, because it's really cool actually. it's
Yeah, that's interesting. So do you think that, like, because soldiers go through this kind of training to become like a collective unit and, you know, I don't know, bond together, maybe in a higher level than firefighters do, that could be protective?
Yeah, like when we looked through huge data sets from interviews with Holocaust survivors, like I did a resilience study afterwards.
Mm-hmm.
That was the most significant part, that in people that kind of always saw, like they always talked about we went through this, and were the people that kind of had then a kind of regular life, not that they never had issues again, like,
Mm-hmm.
but had the family, jobs and stuff
Mm-hmm.
like that. You look at
Right.
the reports when they talked about the trauma and everything, they always talked about we went through this, this happened to us.
Mm-hmm.
And it's a huge difference. Like it's the most significant one actually, and
Wow.
all the other word categories. That's why then the soldier told me yeah, we go through this training and we get trained away to see us like
Mm-hmm.
separate group.
Yeah.
Yeah. So,
Mm.
anyways, interesting. I want to open up the questions to other people too. Please flash your microphones, everyone. And yeah, Serena.
It's a really fascinating talk, and particularly that for the neuromelanin accumulation as a marker of just sort of overactivity.
Mm-hmm.
I've been interested in looking at glial and specifically astrocyte involvement. I'm wondering, have you looked at that, and is this accumulation in part due to a failure of clearance by the astrocytes, or I don't know if you've looked at that.
Well, it's all happening in the cytosol, so I certainly wouldn't rule out that they could impact that, but I guess it would be a little bit further removed, maybe.
Because in reading many of the pathologies that you covered,
Mm-hmm.
I see there's astrocyte involvement, and I'm still sort of puzzling through that. Is there? But here your study was quite focused on the locus coeruleus and the signal of the accumulation of the neuromelanin that even predated the tau accumulation. Was that correct?
Well, I don't know. Well, in terms of the locus coeruleus, the idea is that it's accumulating tau, relatively early, 20s or 30s of age.
Mm-hmm.
But the neuromelanin, it should be detectable in the early years of life. In humans, like 4 or 5 years old, there's some detectable levels.
Mm-hmm.
Um, so yeah, and it's not actually there in other species, like it's there in primates and some other large mammals, but it's not there in rodents, or very little. Like
Oh,
really
that's
old
interesting.
rodents might have a little bit, but
Mm.
yeah. So one factor that it makes you wonder what that's about. Like, anyways, for instance, the VTA doesn't have much neuromelanin, so that's why it's kind of a notoriously hard structure to see. Whereas the substantia nigra is black, and you can see it.
Mm-hmm.
But they're both dopamine neurons, and the reason for that is most likely because the VMAT expression is higher in the VTA, so they're clearing the dopamine faster and sucking it out of the
Mm-hmm.
cytosol, so it
Mm-hmm.
can't much of it doesn't stick around long enough to make neuromelanin. But that makes you wonder, why isn't that happening in the nigra? If this is a dangerous process, this biology somehow has decided to run that risk. There must be some advantage to having neuromelanin floating around longer. I don't know why.
Oh, that's interesting.
Yeah.
And is the neuromelanin, is it just in neurons, or is it in other cells within the brain as well?
Yeah, I don't know if there may be some small amount of it that floats around. I don't know. I mean, I would expect most of it's in neurons and mostly in the catecholamine neurons.
Mm-hmm.
The levels elsewhere are very
Yeah,
low.
that would make sense. It's in essence a metabolic product, right?
Mm-hmm.
Mm-hmm.
Yeah, and it gets wrapped up in these granules right away. So I don't know how much of it gets out elsewhere.
Well, it's a fascinating angle, and it certainly presented as a fairly reliable signal for these pathologies.
Yeah, I mean, we know it's useful. Like, that's the good news. I mean, the bad news is we don't understand a lot of the mechanisms, like the physics of it as well. There's a camp out there that says maybe it's not neuromelanin. And, it's true that we don't understand well why neuromelanin would cause these, signal changes. But there's a lot of circumstantial evidence that it is neuromelanin, or at least one of the factors contributing to it.
Mm-hmm.
So yeah, I think it's really promising. I think it's going to be useful, no question. But there's still a lot to learn.
Well, thank you.
Thanks. Who would like to go next?
Yeah, exactly. Dr. Olu, Kyle, you joined the stage. Go ahead.
Doctor, did you want to go first?
Sure. Sorry I joined a little bit late, but I was wondering if there are any studies looking at changes in the MRI marker in association with medication treatment, especially in the psychiatric populations that you've looked at, like the ones with PTSD or other disorders.
Yeah, I mean, there's no definitive evidence, but we've I think there may be a study that looked at antipsychotics or antidepressants. One study we're involved in was in late life depression, and we saw relationships to some symptoms of psychomotor slowing. But what was even maybe more interesting in that paper, some of the people were given L-DOPA as a treatment for late life depression, and and we found a significant effect that the L-DOPA treatment increased the signal. So that again is kind of confirming neuromelanin having a role, although I guess something else could have changed it too. But we know that a drug like that will boost the catecholamine levels in the brain, in the neurons, and that will lead to increased neuromelanin formation. Maybe it was surprising that even a short treatment had a statistically significant increase, but that's the best example, I think, of a treatment changing the signal and in a way that made sense too.
Yeah, that's very cool to hear. Was this with Brett Rutherford?
Sure was. You know him?
Oh yeah, he's a friend of mine. So
Okay.
very cool. Thank you.
Yeah.
Well, you both are very cool, as far as I reckon. I'm curious about the phasic and tonic patterns of the locus coeruleus, however you pronounce it, and I guess that's like a tonic or burst. And I'm just wondering if you could talk a little bit about that because I'm just kind of curious about that as it relates.
Yeah, I don't know that there's any real data like how different types of activity patterns would lead to neuromelanin formation or not. Is that sort of what you're driving at?
Just in this
Or more
specific
generally?
region, just specific to the region in the brain, generally like how that works, I guess. So just looking for some information, whatever comes
Yeah,
to your mind.
I mean, I don't know much about the electro, the firing patterns in the locus coeruleus. So yeah, I know a bit about the dopamine system, but I can't assume it's the same there.
Okay. The reason why I ask is because of the norepinephrine and the arousal attention states and behavior modulation. And then I kind of think about factors such as like eustress, agency, growth, um, attention, awareness, even how we learn,
Yeah.
kind of also like a little bit of a metacognition.
Mm-hmm.
So the reason why I'm asking why it's firing is because I've always been interested in this tiny blue spot in the brain, and
Yeah.
you're the first person I've heard speak about it on Clubhouse. So thank you very much.
Okay.
Amazing presentation. Greatly appreciated.
Yeah, thanks. Yeah, there's it's a pretty hot part of the brain these days. There's a ton of work, and used to feel like it was kind of like the poor sister of dopamine or something, but I feel like there's a lot of exciting work going on, and people just really focusing on the locus coeruleus and norepinephrine system, and it seems it's relevant for so many things. So yeah, I think there's a lot to learn for sure.
Thank you so much. And I really appreciated your empathy during your presentation. I just thought that I should mention that. And yeah, I'll stop talking, but I
Yeah,
appreciate
yeah,
your
thanks.
time and energy on this very much.
Thank you. Yeah.
Okay, I guess I'll go next. First, thanks for the talk. When you were discussing the advantage perhaps of one of these processes, I what came to mind was rheumatic sequestration. So it could be an emergency sequestration measure
Hmm.
for some sort of pathogen, or the
Yeah.
beginnings of it.
Yeah, I think so. You're asking how does this neuromelanin getting cleared or
Well, you had
being
mentioned,
created?
if I didn't hear you wrong, that it could be um, like, what is the advantage of neuromelanin?
Right.
Was the question you brought up, and I thought that it
Yeah.
could be an emergency sequestration measure.
Yeah. Yeah. So there, that my colleague in Italy who we've been working on with all this stuff, he's kind of like the neuromelanin guru of the world. Like he's devoted his whole career to it, and a lot of his work kind of poses that question: like, is this a good thing? Is it a bad thing? And the answer is, it can be good, it can be bad, depends on different things, and it's certainly serving a function when it's there. Um, one function it does mop up a lot of stuff. It binds metals. That's part of probably why it could affect MR signals.
Hmm.
And it binds other toxins. And it just, so, these granules are kind of full of a lot of bad stuff. So if they ever get broken, that's bad news for this tissues
Like
cells.
via concussion or something, perhaps?
Or whatever is happening in Alzheimer's and Parkinson's that's leading to some cells getting damaged can kind of spiral, right? Then once damaged, it releases that, and that can harm adjacent cells.
Fascinating. I imagine there might be lead found in those sometime.
Yeah, I would imagine I should ask my colleague.
There was a study out of London that had done necroscopy on multi-center. It was multi-continental even, and they were able to find some implication for that in similar process as Alzheimer's. But I was curious how easy it is to run an NM-MRI.
Yeah, yeah, that was. I think I forgot to talk about that. Like that's one of the most biggest assets of this method is, there's different ways of doing it, but there's at least some ways that are really easy you can do just out of the box. Like, it takes usually 7 to 10 minutes, and it doesn't require any special contrast agents. It's just a standard sequence. You have to pair it with a anatomical scan to use it to best effect. Um, but yeah, it's really easy, and so it could be done on any scanner, and has a lot of potential for clinical. It's easy enough to be done in a standardized way in a clinic without some expert needing to be involved.
And we don't need to deploy any new equipment. This is available
Yeah,
technology that's deployed already.
yeah, yeah. There's a version. One of the ways of doing the sequence, you might need a developer sequence. But it's debatable whether that version's even that much better.
Very cool. Really appreciate your answers. Thank you so much. I'm going to pass the mic to whoever would like to flash their mic next.
All right, thanks for your question.
So just a quick point. It's really just the impression that I've gotten. But melanins in general, and presumably the polymers of dopamine and norepinephrine, those are oxidative or can be oxidative polymerization products. So
Yeah.
I inferred that this was either a response to or a mitigation mechanism for oxidative stress.
Yeah, exactly.
Possibly both.
Yeah. That's probably, that's the most obvious purpose. I mean, whether it serves other purposes, but yeah, it's like there's, I guess, what's this, quinones or something that get created from dopamine when they're oxidized, and those are very bad chemicals. So they eventually turn to neuromelanin, get packaged up, and yeah.
Eli, did you have any further questions? Otherwise,
Just that comment, though I'll add like just an interesting little tidbit. Apparently, the melanin in fungi lets them utilize energy from gamma radiation.
Oh, wow.
fascinating. They also synthesize vitamin D via the same pathway that exists in humans. But But who would, Katerina, did you have a question, or do we want to go to Victoria or Jamie? Who would like to go next?
Yeah, I was about to ask if Jamie had a question. But I think he's away from the phone, maybe.
Yeah,
There we go.
Oh,
hiya, I'm here. Doctor, thank you for an incredible talk here. You covered a lot of ground. I suppose my question's a largely general one because, as I said, you made a lot of discoveries and observations here, and I'm wondering, um, has everything you've discovered so far led to you finding more questions that need to be answered, or do you think as well that this work you're doing is helping to fill in a lot of the gaps we're having with everything from being able to predict mental illness to possible treatments or even understanding Alzheimer's, or are we still have a long way to go?
Yeah, I mean, certainly, the more questions you ask, or more questions you address, the more questions arise. And you know, so even if you do answer some questions, it doesn't mean new ones don't spring up. Like, for instance, we could be using it to understand the brain, or we could be using it to help treat people. So if you're just using it to treat people, the good thing is you don't need to know why it works. Just if you see it's related to something, the signal predicts whether you respond to drug A or drug B. That's great. You can exploit it. But to understand what is happening in the brain, yeah, it helps. But then there's always more questions arise. For instance, in this study I'm doing across the lifespan in healthy people, which is very interesting to me. I kind of wonder if the rest of the world's going to be as excited about it. But we're collecting, actually, there's two versions of so-called neuromelanin imaging, two sequences that aren't even that similar, but the experts say that they have the same special sauce that, makes them basically measuring the same thing, even though the sequences are different. And they seem to be in good agreement if you look at, the signal across all the participants in the study with sequence A or sequence B. But, if you look a little closer, they aren't always in agreement. And, one version seems to be picking up on something a little different. But then it's kind of scary to bring that to the world because it's like, you're raising a lot of questions and, it kind of might even undermine some of the work you already did. So it's like the closer you look at something, the more complicated it always seems to get. Short answer.
Yeah, thank you very much, and thank you very much for this incredible work that you are doing. This is incredibly good for humanity as a whole, and I really appreciate your efforts here. Thank you very much.
Thanks so much for the kind words.
I did have a follow-up question, and there was a previous speaker that went through some developmental studies,
Mm-hmm.
and it was about a balance of pruning synapses with through the microglia at critical development stages,
Right.
and the general picture was that and this was done in mice, but the mice are born with too many, and they have to prune to a certain balance for appropriate level of sensitivity.
Mm-hmm.
And if you inhibit that pruning, they're more anxious, so they're less resilient. I'm wondering, because you mentioned that certainly in the Alzheimer's cases that there's a movement to, you can identify the biomarkers. But I'm curious in this case for variations within the population that do have anxiety issues. Do they have a more pronounced signal with the neuromelanin?
Mm-hmm.
I mean, is that a
Yeah.
way to identify them in a sense?
Yeah. I mean, all these things, there's your sensitivity and specificity is not going to be perfect. If you look at group averages, there's been one or two studies showing that people with anxiety symptoms have higher signal in the locus coeruleus, and that's, I guess, PTSD, anxiety like too, and we saw that. So yeah, there could be things happening with anxiety. I mean, I'm really interested in the developmental question too. Like I said, for that healthy lifespan study, mostly I was interested in the younger people, especially for instance, the dopamine system. You know, we know there's so many dopamine disorders, and you know, many of them have a developmental aspect. Um, and so I was kind of curious. Maybe we'll see some kind of clue that you know the signal is supposed to be accumulating gradually as people are you know growing up from children to adults, but maybe there's certain critical periods when it's changing in a different way. Like maybe it's certain kind of flat and then it spikes up at a sharper rate. Like for instance during puberty or something, or maybe there's changes that happen, you know, in later adolescence, which is when people get schizophrenia. And the dopamine system, like you know, I'm intrigued to find those kind of clues, and we'll look at certain symptoms of, you know, even though they're healthy people, we'll look at you know low-level psychopathology of different types, including you know substance use, um, low-level psychotic experiences, and just general psychopathology and ADHD type symptoms too.
Good evening, Clifford. This is Amy. I have a question.
Hi.
Yeah, this is just a general question because I came in late in the room and I don't know if you've already discussed it. Based on the function of the neuromelanin, is it something that can be chemically induced to assist when it's decreasing in level?
Well, I don't know that it would be useful to add more neuromelanin. There's even people who think it's risky to have a lot of it, and that the neuromelanin itself, like high accumulation that happens late in life, could be a risk factor for some disorders. Some people dispute that, but in any case, I don't know that you know it may be helpful in certain circumstances to have some neuromelanin. Like it's like I said, it could be absorbing some toxins, but I don't know if anyone's ever speculated that low levels would be a risk factor and that that kind of intervention could be helpful.
Uh, was there ever a study based on, how black people have more melanin? Is there a
Hmm.
study that there's an increase or a decrease in different races? Is there
Yeah,
one?
that's actually been something that people have kind of raised as a thought or a possibility, and I don't know that it's ever been adequately addressed. I don't know that there is a strong a priori reason to think that. Like my colleague, who's the neuromelanin guru, says that that's a non-starter. That the melanin in your skin has nothing to do with the neuromelanin in your brain. I don't know if he's right. I mean, until you look, I guess you don't know. And it, you know, for instance, in my study in healthy people, we want to look. You know, part of the reason for the study is if this is going to be used clinically, we have to understand what is the baseline, like what is the normal level of neuromelanin for a certain person and what factors influence it. We know that age is a huge influence on it. We see even sex has an influence. We don't understand it well enough yet. When we have more people, we'll look at it more carefully. But yeah, race could be another thing, or ethnicity. You know, different factors that could impact the signal, and we would like to get to the bottom of that.
So, as you get older, do you have an increase or decrease of neuromelanin?
Well,
I
you
just
should
want to
have.
make sure I
Yeah,
understood.
yeah, it should increase progressively, but you know, it depends how you measure it. There should be more in each neuron as you age, but then, you know, at some point when you get older, you start losing some neurons. So, you know, the total amount might start decreasing a little bit because even though individual neurons are gaining more, there's some neurons dying, and at some point the balance may turn to a net loss of neuromelanin.
So, can you explain to me again how does L-DOPA play a role on this?
Yeah, so you know, chemicals that are similar to dopamine or norepinephrine, these catecholamines, they can get eventually converted to neuromelanin. So something like L-DOPA, you know, is one of those types of chemicals. So, you know, if you give it to someone as a drug, um, most likely it would increase neuromelanin formation. And, you know, it's been shown in some, not in the most direct way, but there's indirect evidence certainly that that's going to happen. Whether that's a bad thing, it may not really matter. I don't know if it would have any effect on a person's health if they have a slightly increased neuromelanin accumulation rate.
Because I know that L-DOPA is used for Parkinson's. So
Hmm.
for Alzheimer's, is that something that they're trying to use already too?
I don't believe so.
No?
The
Okay.
dopamine system seems pretty preserved in Alzheimer's, surprisingly. I mean, not, when you get sick enough, it'll start to be affected too, but it's not one of the first things to go there.
Okay, thank you.
Yep.
Yeah, thank you so much. We've been going over an hour and a half, so I wanted to give you the opportunity to basically get some
Okay. Okay. Well, I could take a couple. If there's more questions, I could talk
Okay.
a little bit longer.
Okay. Yeah. Does anyone has more questions for um, Dr. Cassidy right now?
I am curious about these studies involving regular sampling of groups of individuals' lives and different and looking back at the effect of whether they went through something traumatic, had a really bad year, other
Mm-hmm.
types of things, whether that spikes or really follows. Is it really an indicator of in essence just how much total activity there's been?
Mm-hmm.
You mentioned that.
Yeah.
Oh,
Well,
go ahead. Go ahead.
no.
Well, you mentioned you were involved in a study of healthy people. Are there other of these types of, do you call those longitudinal studies?
Yeah. That in this case, it is longitudinal study. Yeah. So we would like to look at that. Like right now when I talk about, I'm just these are cross-sectional studies looking at age. Seeing that older people have more neuromelanin. But if you look within subjects, you'll get more information, right, to see how one person changes over time. And that would be pretty exciting, especially, if you look at adults, maybe the rate of accumulation is lower and it may be noisy, but especially the young people, when they're accumulating at a high rate, you could see a lot of change in signal relative to the noise, and maybe more subtle variations are detectable and maybe related to life experiences. We'll see if any of this pans out, but we're interested in looking at, like, for instance, in the dopamine system, people are sensitive. The dopamine system's sensitive to social success, right? Like people with high socioeconomic status tend to have higher dopamine levels, and monkeys that move up or down the hierarchy, their dopamine is supposed to shift. Um, so, like, we'll look at kids that have a big social network. Are they accumulating neuromelanin at a higher rate? If they're just maybe more generally just having a rich life experience, for instance, we look at like engagement in different activities, sports or music or whatever. Does that, affect it versus kids that are just sitting on the screen all day where you're looking at screen time too? Is that understimulation or overstimulation? There's a lot of talk out there in the media about, screen time being bad for kids, for instance, but there's no data. Like people say, oh, screens are hacking your dopamine system, but you can't study the dopamine system in kids, so it's just talk, so hopefully we'll see some clues.
Yeah, I have a couple of questions, if that's okay.
Yeah.
So one is, since neuromelanin seems to be increasing with age, does it correlate with like methylation and epigenetic changes, and could that be kind of a way to control neuromelanin if it's kind of a causation and not just a
Mm-hmm.
symptom? Is there any study or thought in analyzing that?
Yeah, I mean, I don't know. I mean, we're collecting DNA in that study, and we're getting collaborators to help because that's not my area of expertise. So, but I don't know that the accumulation with age is a bad thing. Like, there's that school that says it might be, but I don't know that we have enough. I think we're far away from enough confidence to say that we should intervene to stop it, because for most people it doesn't seem to be that bad. Like a lot of people live to be 100 and they have a ton of neuromelanin, and it doesn't seem to be causing that much harm. And so it must be, if it is bad for you, it must be an interaction with some other factor that makes it bad, would be my
Yeah,
guess.
I was thinking that maybe, there are some epigenetic changes that causes, and then you could detect
Hmm.
it in blood also instead of just imaging.
Yeah, I think it would be really cool to look at it with epigenetics. And my colleagues that were looking at that, but isn't blood better than saliva for epigenetics?
Well, saliva would be easier, right? So
Yeah, well, that's the thing. We're collecting saliva, but I know there's some things that saliva is not good enough for.
I mean,
Anyway.
many people go. I was also thinking of going to saliva because it would just be less kind of a hassle.
Yeah, no, that's why we're using saliva and not blood, especially with children in the study and stuff. We don't want to scare anyone off. It's already hard to get those people. Um, but I know that there's some limitations that even if it's easy, it's sometimes not as powerful.
And I know that there's some. I read about the study that is looking at neuromelanin in the periphery, like in the peripheral neurons, and
Hmm.
not sure if exactly the same, but could it also correlate um, and be also like a easy, accessible way of looking at neuromelanin increase there too? Or do you think it's very, it has to be in these specific brain regions?
Yeah, I don't know about how it would be acting in the periphery. I mean, I guess maybe it's the same pathways that are generating it from catecholamines in other parts of the body. But I would imagine the levels would be low.
Yeah, interesting. No, I always just think of, cheap scaling ways of maybe, population that don't have access to imaging and stuff
Oh,
like,
yeah. Yeah, maybe you could capture some of the same processes. I don't know. Yeah.
or maybe in the eye, there was,
Mm-hmm.
I'm not sure, there was a recent study that looked at retina, and they could kind of see neurodegeneration in the
Mm-hmm.
retina at the early diagnosis. That's
Yeah. I
maybe
don't know
they.
about that, but they do have, there's a way of doing sonography of the substantia nigra. I don't know how it works, but, um, I know there's been studies and it actually is useful for some applications. So that would be cheaper, I guess, but maybe not as high quality a measure in the end. I'm not sure.
Yeah. Well, interesting. And again, thank you so much for coming and answering all of our questions and giving us such a amazing talk about your research. I learned a lot, and yeah, I hope. And thank you everyone for coming, asking great questions. I see that Brandon and Kyle, they shared a lot in the chat. Thank you for that. And yeah, I hope maybe you'll come back one day with some research updates.
Okay.
Um, we wanted also to plan in the future to have like an expert round, maybe like roundtable discussion with guest speakers that came and kind of overlap in their
Sure.
research, maybe.
Yeah.
And yeah, I hope you enjoyed it. I
Yeah,
know
it
it's
was great.
been Clubhouse, so
Yeah. Thanks so much for inviting me. It was really fun. And yeah, I'm going to stay in touch and check out some of the other talks.
right. Amy is using the new feature of um, clapping and showing like emotions. I'll do that now too. Okay. Thank you,
All
everyone, and have a good night. Enjoy your holidays, like going
Yeah,
back. And
will do.
thank you, everyone. Yeah, follow the club if you like this type of discussion. We'll have tomorrow Dr. Vogt from Germany joining us and talking about brain signal pathways that control intake. And then we'll have Dr. Campisi talking about CD8 T cells that can maybe characterize ALS. And yeah, we'll have many more talks that will be really interesting. Dr. Liang, he will talk about deep sea virus, his deep sea virus discovery, and how it can explains a little bit more about the origins of complex life. So yeah, join us again, and I hope to hear you all soon. And thank you, Cliff, again. It was a great honor, and enjoy the rest of your summer.
Okay,
Okay.
thanks again.
Thank you. Thank you
Thank
Thank
very
you
you.
much,
very
Doctor.
much.
Thanks, everyone.
Enjoy your time. Bye bye.
Good night,
Bye
everyone.
everyone.
Good night.
3, 2, 1. Bye, everyone.